My dear students, in my previous class, it was taught that the invertebrate are 10 phylum. Porifera, cnidaria, ctinophora, platyhelminthes, nemathelminthes, annelida, arthropoda, mollusca, and echinodermata, as well as hemichordata. All these are non-chordate. Chordata actually true, notochord is absent here. They are invertebrate. They are very simple organism and they are primitive. Today we shall discuss about the salient features, the characteristic features of the phylum Porifera. Porifera is the animal which is their normal habitat is aquatic. Mostly their habitat where they live they are mostly marine. That means they live in the sea water and few are fresh water. Few fresh water. They inhabited few fresh water. Their body organization, which is the type of their body? Their body is very simple. Second is the body organization. Their body organization is very simple and this is cellular form of organization. Cellular form. They are multicellular, but these cells are not well organized fashion. Ectoderm, endoderm and mesoderm, these different types of layers are absent. These are some sort of random fashion these cells are arranged. Their cellular form, their body symmetry, they are asymmetrical. They are, when we cut their body in a vertical plane, each segment from which we can get will not similar with each other. This is the body symmetry. Is asymmetrical. As their body is very simple, so they have no phylum. Phylum absent. Phylum is absent. Their body structure, when we consider their body structure, first we draw a specimen, that is the cycon. Cycon is the one of the member of the phylum Porifera. These are their structures, example of cycon. This cycon have a large opening. This is a large opening. This large opening is called osculum. And in their body, many small openings are also present. These small openings are called ossotia. Osculum and ossotia. These are the external features. Within their body, a cavity is also present. This cavity is called spongocoel. This cavity is called spongocoel. Another feature is that they are in their body, the skeletal, a calcareous skeleton also present. This skeleton is made by calcareous spicule or spongin fiber. This is some spicules also present in their body. These spicules make their body skeletal structure. These are the most important salient features of Porifera. That means their body surface, body has two openings, has two types of opening. One is called osculum, large. This is called osculum. Its number is 1. And many, small many pores. These are called ostia, also tia. What are the function of these two types of pores? As these specimens live in the sea and live in the water, the external water enter into these small pores. As their body is porous, small pores, that is the ostia, are present. Through these ostia, external water enter into their body. And these water ultimately reach into this cavity, spongocoel. Then this water come out through the body by means of osculum. That is the osculum. Through ostia function, water enter into body, and osculum, that is the function of osculum, water come out. The water contains different types of microscopic organisms. These microscopic organisms are, when water enter into this canal, spongocoel, these microscopic organisms are digested by some special cells in this present in this canal. This canal is called spongocoel. That means body canal is called spongocoel. Is the structure. Then their body surface skeletal material, this is called spicule or spongin fiber. Body skeleton contain spongin fiber. These fibers provides their skeletal structure organization. Then a special type of cell is present here. This type of cell is called choanocyte. Choanocyte cells are present here. These cells and the spongocoel. Water enter into this spongocoel and the choanocyte are present. These cells helps the, what is the function of the spongocoel? Choanocyte. These cells helps the intercellular digestion. After digestion, as these cells are present in this area, after digestion, the canal system, after digestion, the excretory substance are expelled in this cavity and come out. This area also helps in excretion, also helps in respiration. This type of two functions are performed by this cavity spongocoel. Their male and female body are not differentiated. Single body contain male and female reproductive structures. That is why the specimen is called hermaphrodite. Hermaphrodite. What do you mean by hermaphrodite? That means no clear differentiation between male, this symbol denotes male, and this is plus sign. This is symbol denoted by female. Male and female organ are present in the same body. That is why these are called hermaphrodite. And their development. After fertilization, what develop? Zygote develop. This zygote differentiate and form a mature animal. During their developmental formation, during their formation of the adult, some larval stage are present. Larva present. These are the indirect development. When larva is present during their life cycle, indirect development. And the larva is called amphiblastula and parenchyma. Amphiblastula and parenchyma are two types of larvae. Amphiblastula, parenchyma. These two types of larvae are present during their developmental process. What is the developmental process? In which stage? From zygote to the adult stage. Some larval stage develop, transient larval stage. These larval stage are named as amphiblastula or parenchyma. Example of Porifera. Several types. There is one is this is the picture is called Cycon species. Euspongia and Spongina. Bath sponge, Euspongia. Euspongia and Spongina are two genus under this phylum. Bath sponge in some structure, bath sponge and in some cases it looks like when we look front in a erect position, this shape develop, and when it is present in a reverse way, this structure we may see. Another spongilla. Spongilla is another structure that have very irregular in their shape. This is called spongilla. And this is bath sponge or Euspongia. These three important genus are very predominant in the phylum Porifera. Next, we shall discuss about the two phylum, that is the cnidophora and cnidaria. Cnidaria and ctinophora. Then we discuss about now today's topic is cnidaria. Cnidaria. Phylum cnidaria. Cnidaria is the more developed phylum than Porifera. Here, their body organization. First, we consider their body organization. Body organization. Cnidaria body organization, when we consider cnidaria, their body consists of two cell multicellular layer. Two layers of cells. Two layers of cells present. These are called ectoderm and endoderm. In between an ectoderm and endoderm, mesohyl is present. This look like, suppose this is a outer layer. When we divide into some cells are arranged in circular fashion. This is the outer layer. This is the ectoderm. Inside the ectoderm, the inner layer is endoderm. This is the endodermic cell layer. In between ectoderm and endoderm, some cells, some jelly-like substances are present. These are called mesoglia. This is mesoglia. Body more or less similar. Their symmetry. Second is symmetry. Their body symmetry, when we look, their body symmetry is radial. Radial body symmetry. That means when we dissect this body along a plane that pass through their central axis, and this plane may vary, they present in different angles, then we get different body parts. Each body part are similar with the other body part that originate during the longitudinal dissection. These are the radial symmetry, and also this body is acoelomate. Coelom generally absent. This type of organisms show two types of their form. One form is the polyp, and second and medusa. Polyp and medusa, two type of developmental forms present in this phylum. In some cases, polyp converted into medusa and medusa converted into polyp. This conversion generally takes place in their life cycle. This conversion is called metagenesis. What is the polyp? Polyp is this type of structure, a sea anemone. When this is the sea anemone. Adamsia. Sea anemone is called the Adamsia. It is a polyp. And another member is called Aurelia. This is the Aurelia. Adamsia and Aurelia. Aurelia is the medusa. It is a medusa form. And this is a polyp form. Polyp is sessile. It has no capability to move and reproduce asexually. That means they do not form male and female reproductive unit. Fertilization generally takes place in this organism. But in Aurelia, there is a medusa stage. It is motile, can move and sexually reproduce. Fourth point, their body contain a water vascular system. Their vascular system is water mediated water vascular system. And the water vascular system, the general cavity is called the gastrovascular cavity. This is called mouth. Now it opens into mouth or hypostome. Mouth is called hypostome. This is mouth called hypostome. And body called a central cavity. This cavity is called gastrovascular cavity, the hypostome. And some around the hypostome, some small finger-like projection is called. These are present. These are called tentacle. These are called tentacles. That is, mouth is surrounded by mouth or hypostome is surrounded by tentacle. For defense and capture prey, tentacle they play a very significant role. In this tentacle, a special type of defending cells are present. These cells are called cnidocyte or cnidoblast. Cnidocyte or cnidoblast. These cells helps the organism to capture prey, and it also helps them prevents themselves from external prey. This cnidocyte or cnidoblast contains nematocyst. Nematocyst present within the cnidoblast. When we look this structure, this is the cnidocyte. Within the cnidocyte, this type of structure is present. This is called nematocyst. This structure is called the cnidoblast. they during their life cycle they do not show any larval stage. That is, their development is more or less direct. And the digestion, they follow two type of digestion. That is the intercellular as well as extracellular digestion. Two types of digestions these organisms follow. Different members, cnidaria, in the cnidaria, different members are present. They are nicknamed sea anemone, sea pain, sea fan, coral. One of the members is coral. Coral is a solid organ body made by calcium carbonate and different colors present in their body surface. These are the coral medrina. That is the sea anemone. Sea anemone is called Adamsia. Sea anemone. Different examples are sea anemone, sea anemone, sea pain, Pennatula. Sea anemone is the Adamsia. Sea pain, Pennatula. Sea fan, Gorgonia. Portuguese man of war, Phycelia. Another member is Phycelia. Portuguese man of war, Phycelia. Brain coral, that is called the medrina. Different types of all other different genus belongs to this phylum. Now, next, the phylum is ctinophora. Ctinophora and cnidaria together called Coelenterata. Cnidaria plus ctinophora. Together called, previously called Coelenterata. Ctinophora is the primitive member. Their body, the first character is similar. Their body organization is very simple. Their body organization is a tissue type, tissue-like, just like cnidaria. Their body also contains two layers of cells. One is the ectoderm and the endoderm, and in between ecto-endo, mesoglia is present. Their symmetry also radial. When their body is vertically, we dissect this body in different plane that pass through the axis. We get different body parts, and each part is similar with the other generating body part. This is the symmetry. Here, all these structures generally are absent from this. Now, their locomotor organ is 8 ciliated part, 8 ciliated area called complet. Some members of this phylum show, that is the ctinophora, show bioluminescence. That means they emit some special type of radiation when we illuminate it with UV light. This is a bioluminescence property. Example, Tinoplana and Pleurobranchia. Dinoplana, Pleuroranchia are two genus under ctinophora. Ctinophora, also called sea, the total phylum is called sea walnut. Now, in the next class,